Heart attack. It's a deadly killer. A leading cause of heart attack is thrombosis, the formation of unwanted blood clots in a blood vessel or the heart. Blood clotting is a carefully regulated biochemical system employing a large cast of interactive proteins. It's a cascade that is normally held in check until a bleeding injury fires it into action. Our heart attack victim, however, has a problem with his blood clotting cascade. Something has triggered it inappropriately, and a series of associations or complexes are forming between clotting proteins. What can we learn about the proteins that initiate the clotting cascade so that we can intervene when their behavior goes awry? We'll begin with a look at two proteins whose association triggers the blood clotting process. Using a laboratory technique called Sds Polyacrylamide gel electrophoresis, we'll determine the molecular weights of these proteins. Next, another technique called Analytical Ultracentrifugation will help us characterize the way these proteins combine. Finally, what we've learned about the structure of this protein complex suggests strategies for keeping inappropriate initiation of the clotting cascade in check. Let's meet the molecular culprits responsible for that heart attack. The coagulation enzyme called factor 7A and an enzyme cofactor called tissue factor, or TF. They combine to form TF7A.
Well, this is a molecule that's involved in initiating blood coagulation. It triggers. It's one of the trigger molecules at the top of the cascade. And once that complex forms, it can just set the whole cascade going.
Since clotting requires the proper association of 7A with its cofactor TF, we want to understand as much as we can about their structure and their binding mechanism.
There are a lot of things you want to know about this complex. One of the things you want to know is where is it in the body? You want to know what does it do, what does it act on, and you want to know what controls its level of activity.
In the body, TF is firmly planted in the cell membranes of vascular tissues, and that makes studying it a bit tricky. Fortunately, a synthetic version of TF that is soluble in water has been developed. This protein is called soluble tissue factor, or STF. We can use STF to learn something about the way TF binds with 7A on the surface of cell membranes. Here's what we want to discover. How many molecules of STF and how many molecules of 7A combine in a clotting complex? In other words, what is the stoichiometry of the complex? What are the shapes and sizes of the two kinds of subunits? How are 7A and STF arranged in the complex, and how strongly are they held together? The answers to some of these questions can be found directly and easily by measuring molecular weights. So let's begin our investigation with a powerful laboratory technique called Sds Polyacrylamide gel electrophoresis, or Sds Page.
Sds Page has a lot of uses in biochemistry. What we're going to do with it here is determine the size of the chains and the proteins that are making up this STF7A complex. We need to know the first piece of information, how big is each chain, before we can determine how big is the complex where we have many chains together.
Here's the setup for Sds Page. A polyacrylamide gel is cast between two glass plates separated by spacers. After the polyacrylamide solution is poured, a comb is inserted into the top of the sandwiched solution. When the gel has set and the comb is removed, it will leave a neat line of wells into which different samples can be applied. The gel is mounted between two buffer chambers. An electrode is in contact with each chamber, one positive and one negative. So when voltage is applied, an electric field is generated across the gel. Charged molecules will move in this field toward the electrode of opposite charge. Let's see what this phenomenon, called electrophoresis, can tell us about 7A and STF. First, a sample of STF-7A is heated with sodium dodecyl sulfate, or SDS, an ionic detergent commonly used in dish soaps and shampoo. This treatment disrupts the bonds that stabilize a protein's normal three-dimensional structure and unravels it into an elongated strand. The detergent molecules bind and give the proteins a net negative charge as well. The treated protein sample is gently loaded onto the gel using a fine-tipped pipette. At least one well is loaded with protein standards, a mixture of proteins whose molecular weights are known. A small amount of tracer dye is added to the samples so their progress through the gel can be monitored. On goes the voltage, and the negatively charged proteins begin migrating toward the positive electrode at the bottom of the gel apparatus. The polyacrylamide in the gel forms a tangled network of spaghetti-like strands. This matrix acts like a sieve, slowing down larger objects more than smaller ones.
Everything sort of requires some prior knowledge or prior experience. You can initially, for instance, run a gel and just get a rough idea of what kind of molecular weights you're dealing with. And then you might tailor your next gel so that its porosity is better suited for these samples.
After the gel has been run, each size of protein will yield a band at the position it managed to reach in the gel. A blue dye that stains only proteins helps us visualize these bands. The bands from the sample of known standards will be our yardstick for comparison.
By comparing how those proteins run to how your samples run, you can get a rough idea of the molecular weight of your samples.
Proteins of like molecular weight will be at the same level in the gel. Comparing the migration of STF and 7A to the bands in the protein standards lane, we estimate the molecular weights to be approximately 26,000 and 48,000, respectively. Note that these are approximations. So we've determined the individual or monomer molecular weights of STF and 7A. Now we need to find out the size of the complex that they form.
We want to know the size of the complex for a couple of reasons. One of them is we know that a complex has to form. We know that from other biochemical information. What we need to know, is it just one STF and one 7A, or is it one STF and two 7As? Is it two STF and two 7As? We need to know the stoichiometry of the complex, because what nature can do in terms of the regulation of that complex depends upon how many different chains are present.
To determine the molecular weight of the whole complex, we'll use a different protocol, one that can tell us something about how STF and 7A interact. Analytical ultracentrifugation relies on the fact that particles in solution will sediment if acted upon strongly enough by a force, like gravity.
The reason we want to make the particles move in the gravitational field is we can learn a lot about the size and the shape of the particles as they move in the field.
Particles that are denser than the solution in which they're suspended will eventually sediment to the bottom of their container, like this sand does in water. Sedimentation is caused by Earth's gravitational field acting on the sand particles.
On Earth we have 1G. If we take a molecule the size of STF or 7A, it's unaffected by that 1G. It just isn't going to experience that one gravitational force. So we need to have a lot more. The way we do that is by setting that rotor spinning. And if you take a bucket of water and you spin around with the bucket of water, it pulls out on you. If you spin a protein molecule, it feels the force of gravity. In the centrifuge, we're getting something on the order of 200 to 300,000 times the gravitational force on Earth, and that's enough now to make these very small molecules respond. They move in the gravitational field.
Let's examine what goes on inside this sophisticated device. Protein samples loaded in special containers called cells are inserted in a motor-driven rotor. The motor can be set to reach speeds of up to 60,000 revolutions per minute. At such speeds, air friction would heat the rotor to red-hot temperatures, so air is removed from the rotor chamber to create a high vacuum. Also, since the forces generated can cause the rotor to fragment, the entire assembly is housed in an armored chamber. An optical system uses pulses of laser light to collect data from cells in the rotor. The spinning rotor creates a centrifugal force field, which increases in strength with distance from the center of rotation. Particles move in the field toward the outer side of the cells. Assuming equivalent shape and density, particles of greater mass will always sediment faster than those of lesser mass. This behavior helps us determine the molecular weights of the particles. We'll run an experiment to measure the average molecular weight in samples of mixed STF and 7A. 3 samples, each with a different concentration of protein, are loaded into chambers in a cell. The rotor is set to spin at 20,000 RPM for about 10 hours. At this low speed, particle sedimentation is counterbalanced by particle diffusion in the opposite direction. What we achieve is called Sedimentation Equilibrium, a smooth gradient of protein running from low concentration to high. Concentration readings collected by the optical system are delivered to the computer as a pattern of black and white stripes, referred to as fringes. Here we see patterns for each of the 3 sample concentrations we loaded. The upward shift along the fringes indicates increasing protein concentration. The greater the shift, the higher the concentration. The graphs at the bottom of the screen plot amount of fringe shift against distance from the center of rotation. These graphs give us our working data. Now we're ready to extract molecular weights from the data. First, the range of relevant data is selected by the investigator. Next, a model is proposed, the investigator's best guess at an equation that might properly describe his data. The computer then adjusts the variables of the equation again and again until the model has been made to fit the data the best that it can.
Back in the old days, people took photographs of concentration distributions. Some poor sod then spent days measuring the radial positions on the photographic plate and tabulating it all, doing all those calculations by hand. And apparently many a technician quit in disgust after a short period. The computer does all that in a blink of an eye, but you still have to tell the computer what to do.
Our model was correct, so we are able to calculate an average molecular weight of 74,000 for the samples of mixed STF and 7A. Note that this is the same as the sum of the molecular weights of one 7A molecule and one STF. So we conclude that complexes were formed between 7A and STF, and that the 2 molecules associate in a simple 1-to-1 relationship to make the complex STF-7A. Our experiment reveals something else about STF-7A. The average molecular weight of the particles doesn't change over the range of concentrations generated by our experiment. This means that almost all of the STF and 7A molecules are complexed even at extremely low subunit concentrations. In fact, for the percentage of STF and 7A molecules present as complexes to be significantly reduced, we would have to dilute our samples by more than 100-fold to a concentration so low it can't be measured with our equipment. This tells us that the association between these 2 molecules is very strong indeed.
We now have an idea as to how much energy is going to have to be put in there to break that complex apart. One way of medical intervention would be to prevent that complex from forming. But we need to know up front how much energy are we going to have to put in? What kinds of things are we going to have to put in there to break that complex apart?
If we repeat our experiment using different cells and a higher rotor speed, we can monitor the rate at which particles sediment toward the bottom of the cell. This is known as sedimentation velocity, and it will tell us about the shape of the particles.
Because we know the molecular weight of each of the molecules, 7A and STF, we can make an estimate based on some mathematical manipulations as to how fast they should move if they were perfectly spherical. If they're perfectly spherical, they will move the fastest they can possibly move. If we make them elongated rather than basketball-shaped, more of a football-shaped particle, they will move more slowly because they have a greater surface area.
The sedimentation velocity run reveals that 7A and STF are both rather asymmetric. Interestingly, the length of the complex is greater than the length of either molecule alone. This implies that the 2 elliptical particles do not bind side by side, but instead overlap for part of their lengths. As we'll see, the physical shape of the complex holds implications for its operation in the bloodstream. How can our structural profile of the protein complex STF7A illuminate our understanding of its behavior in vivo?
It turns out that a lot of the blood clotting factors are somewhat asymmetric. And the reason for this may be that the chemistry, the active site of the enzymes, are out at the tip of the protein. And the proteins are now tied down to the surface of the membrane by the tissue factors. So the 7A has the catalytic site. The tissue factor forms a scaffold, a place for this to sit on the membrane. And it's going to do chemistry out here at the tip.
These findings have stimulated much interest in a possible clinical role for STF, the soluble form of tissue factor TF. It's known that for the clotting cascade to begin, 7A must associate with TF, and at the same time be in close proximity to lipid membranes. Since TF naturally occurs bound to the membrane, this condition is always met, and clotting is initiated. While still speculative, the therapeutic idea is this: soluble tissue factor, STF, could be injected into the bloodstream of a patient in whom unwanted blood clotting occurs. Once in the bloodstream, the injected STF would compete with the membrane-bound TF for available 7A. With all available 7A bound to STF, none would bind to TF, and the clotting cascade would not be initiated.
And therefore you could actually prevent blood clotting from occurring when you don't want it to occur.